Let's try to understand about the chick development. Before going into the depth, you must need to know about some sort of the terms here. Amnion, which is providing the protection to the chick, and also chorion, which is important for the exchange of gaseous material. Yolk sac is providing nutrition, and allantois is providing the all waste product to remove from the chick. Okay, this is what present in the chick. Here you can see there are some sort of the parts are present in the chick here. When you see the yolk sac, which is present in the yellowish color, this whole yolk sac have a particular vitelline membrane which is protecting all of the yolk sac, and also in center part there is cytoplasm is region is present, and in the cytoplasm the blastodisc region is present. This is the only part where the development of the embryo will happen, not even in the cytoplasm, not even in the yolk sac. Okay, this is the blastodisc where the whole division and the first single cell will be start develop here only. When we try to understand about the which type of cleavage is present in the chick, there is microlecithal is present, and we are calling it as a megalecithal, or we can also call it as a polylecithal. Okay, here macrolecithal, megalecithal or polylecithal is known as a presence of the yolk. There is huge amount of the yolk is present in the chick, that's why we are calling it as a poly or mega or macro. Okay, and when we are seeing the disicoidal meroblastic cleavage is happening inside the chick, why we are calling it as a disicoidal cleavage? Because there are only these particular blastodisc region only doing the cleavage part. Remaining another yolk sac, cytoplasm or whole egg is not dividing. So that's why we are calling it as a disicoidal meroblastic cleavage. And from here blastodisc we are forming the blastoderm. So this is the blastodisc is later form the blastoderm region, and you can see here one single cell which will divide and cleave and form two cells. Later it will go into the four, and then it will divide and make 8 cells. Later it will divide into the 16 cells likewise. Okay, so till the division of 7 to 8, the all genes that present in the chick is came from the parents only. But after 7 to 8 division, the chick will start providing or expressing its own gene here. Okay, before you can understand it here in the easy way, 7 to 8 division, till the 7 to 8 division, what will happen? The parental genes are working all over the embryo. Okay, 7 to 8 division means 128 cells. Before 128 cells are forming, whole genes are responsible by the parents only. But after 128 cells are formed, then chick will start expressing its own genes. Okay, and here some parts like posterior and anterior, they both will form the primitive strike. With the help of primitive strike, they will form the primitive group. The primitive group is nothing but, primitive strike is nothing but, first you need to know this, it is like one type of the inside going of the all cells. The cells are migrating inside the region of the blastoderm region. Okay, and primitive groove is the region where the complete blastopore is formed in the amphibian side. Amphibian animals where the blastopore is forming. Same like this happen in the chick also. Means blastopore is nothing but anus, right? So primitive strike also forming the anus part in the group. But what is the main difference between the means primitive strike and primitive groove? This primitive streak is a completely depth, means a whole huge hole like structure, but more deeper structure is present in the group. We will see and understand it in the later lecture. Let me explain you more about this. First, blastodisc region is present. This blastodisc is present in the one single cell. It will divide and cleave and form two cells. Like versa, they will be divide and make so many cells here. Okay, after formation of as many as it can divide, they will form blastoderm region. Blastoderm region is nothing but from here to this complete region is known as blastoderm. But when we are seeing only upper region, this region is known as epiblast cells. Okay, remaining part known as a blastoderm, but the upper region part is known, upper layer is known as epiblast cells. And when we try to understand, this is the yolk region present, and here the cavity is forming. This cavity is known as a subgerminal cavity, and the subgerminal cavity is formed by the blastoderm, and this submarginal cavity also taking the nutrition from the albumin only. Also, this blastoderm only forming this epiblast layer also. This is all happened by the blastoderm only. Okay. Here you can see another structure. This is completely epiblast cells. Here subgerminal cavity is present. This is posterior side and this is anterior side. And from the epiblast cells, some cells will be fall from the epiblast cells and they are known as hypoblast cells. This process of falling from the epiblast to the central region, means cavity, subgerminal cavity, is known as a delaminating or delamination. Okay, what will happen next? There are many development, many growth, many division of the cells will occur again, and at the end of the posterior side, many cells will be epiblast cells will be forming, and here is a collar sickle form. Okay, this collar sickle is nothing but thickness of the epiblast cells is known as a collar sickle. From here only streak of the primitive will form, and this is known as a PMZ, means posterior marginal zone. This particular region, posterior side region, is known as posterior marginal zone. Okay, and also from till to there, from here to there, this is known as area pellucida, which is responsible to form the embryo. And also there is blastocele cavities forming. Another area is present here, which is known as area opaca, which is not at all responsible for the formation of embryo. There is no involvement of the area opaca in the formation of embryo. As we have seen, hypoblast cells are formed by the epiblast cells, where it is falling from the epiblast cells. But when we are seeing at the side of anterior side, they are known as a primary epiblast. But when we are seeing in the posterior side, they are known as a secondary hypoblast cells. If we try to understand what is streak, let me explain you. We are having lots of epiblast cells on the surface of the cell, right? There are so many cells are formed. But these cells should need to move inward direction like this. Okay, they all migrated inside and they will start making some sort of the cells. Like why they are moving? Because they want to form germ layer. One is endoderm layer and ectoderm layer, top layer, and mesoderm layer, which is present inside the central region. So this is happening because of the primitive streak. Primitive streak is nothing but the movement of the cell inside like this. Okay, they all are moving, migrating inside. From this region, means posterior region, where the primitive streak will start, and they will continue till the anterior region. Okay, till the here. When they will go towards the anterior region, there will be Hensen node will be formed. This Hensen node is responsible for the gastrulation formation, and it also, as it is same like a blastopolyp, which is present in the amphibian, and you can see here the primitive strike will form and Hensen node is formed here. From here it will move towards the anterior side. And chordomesoderm is nothing but mesoderm cells will form and then they will be converted into the notochord. Collar sickle will form the axis, and gussecoid. Gussecoids are the genes which are controlling